I was really quite excited as I was putting the slides together for this talk, because this is the first face-to-face talk I've actually given in ages, and I realise how much I miss it, because there's nothing like interacting with an audience. So it's really, really great to be here. And what I've put together today is really just to talk a little bit about my history, but what ignited my passion for research. And I'm a person who is easily bored. And the one thing I love the most about my career and my job is that I'm never, ever bored. And to me, that is such a privilege. So I thought I'd give you a bit of a potted history of me. I grew up in Newcastle, which is renowned for its beautiful beaches. I went to Newcastle Girls High School. This is me in the little red pinafore over here with my family, my mum and dad, and this great car. And then after I finished high school, I moved to Sydney to go to the University of Sydney, where I did my medical degree. Now, in those days, there weren't gap years. It was, it just wasn't de rigueur, and I don't think my mum would have let me take a year off. So after going from school straight into medicine, I must say I was suffering from a bit of study fatigue. And so in the third year after my third year of my medical degree, I decided to take time out and do a Bachelor of Science in Medicine, so a full year of research. And as Chris said, that's where I found my passion. I absolutely loved it. I was researching birth defects, particularly those caused by the drug thalidomide. But at this time, and I think showing our age here, I went down to spent a lot of time at the Royal Alexandra Hospital for Children, and again started working with the paediatric pathologists there, but again was very, very interested by looking and trying to understand the basis of birth defects in children. I think the other thing about my career that I want to note is it very much tracks with the technology explosion that we've had over the last couple of decades. So when I did my BSc Med in 1982, this literally, the way we did it was cut and paste, but not in that computer sense. My mum would type out my thesis, and then I would edit it by cutting and pasting and sticking bits together and marking out where the mistakes were. And then my mum would retype that page. At least she had an electric typewriter, so that was quite advanced for the time. I completed my medical degree in 85. And I then I went into paediatric training and then into paediatric neurology training. I'm sort of condensing this down. But I did my doctorate, was very much a clinical doctorate focused on a disorder called neurofibromatosis type 1, which is a neurogenetic disorder. At the time it had come to fame because it was thought that the elephant man had NF1, which it's actually been proven not to be true after that. But having that combination of research and clinical practice to me was something I really always did, even as a student into my training, because it was always a piece of me, something that I was leading and thinking about. And as many of you know, coming through internships, you're doing a lot of service work, but to have something that really inspired me that I could be thinking about was always part of what I did. When I did my doctorate, this is the computer I used. It was with floppy disks, WordPerfect, DOS programming, but it was really great to be able to actually do your research with this computer. We didn't have email at that time, and we couldn't Google things. I think the other thing I wanted to note is how important mentorship is. And so here on the left is Robert Ouvrier, who was the head of the Department of Neurology at the Children's Hospital in Sydney, and really just the archetypal clinician-scientist. He probably worked 150% because he was a full-time physician, but also became a well-renowned expert in terms of peripheral neuropathies. And I did my neurology training and finished my doctorate in Sydney, but then I took up a genetics job at the Harvard Genetics Program based at Boston Children's Hospital. And the reason that I decided to do that is in my work, my doctorate, I was dealing with neurogenetic disorders. In my clinical practice, we could recognise the genetic basis of so many children's diseases, but because there was family history, we could see the patterns that were emerging, but we could only give a diagnosis in less than 10% of these cases. And this newfangled genomics thing was coming out, or in terms of the discussions around sequencing the human genome. And to me, that was going to be the basis of so much of the work into the future. Also, I recognise that probably half of genetics is neurology, is neurogenetics. And so as I was doing my clinical fellowship and then doing my laboratory postdoc, it was really, I was actually quite valuable because I was doing that mix of work, and I sort of became the resident neurologist within the genetics program. It was there that I worked with these two great scientists, Lou Kunkel, who discovered the dystrophin gene, which is responsible for Duchenne muscular dystrophy. And I worked in an environment where everybody on the floor in our building, Enders 5, was working on different aspects of neuromuscular disease. And this was one of my other mentors, Alan Beggs, who really taught me everything I know in terms of the laboratory work, and we still collaborate today. So mentorship is incredibly important. I was based in genetics at this place, the Children's Hospital in Boston, which has a very rich surrounding in terms of that research environment. And one opportunity I was given as part of my genetics fellowship was to go to this amazing two-week series of symposiums and workshops based at the Jackson Laboratory, which is in Bar Harbor in Maine. And this guy up the top had was started to work on this newfangled thing called the World Wide Web. And I'll never forget, we had clinics every afternoon, and we had mouse clinics where we saw all mice with genetics mutations. We actually met with series of patients in terms of difficult diagnoses. But then one afternoon we had a computer clinic, and we were introduced to this thing called the World Wide Web, and it's thought that it might be useful in the future, particularly in relation to genetics research. And my God, has it been. I then came back to Sydney, and again, it was, I got a fax from my mentor here, Robert Ouvrier, saying there's this position, they're creating a new type of position called a career scientist award, it was like a career development award, really aiming at people that were doing clinical work but wanted to do research. And this was back in 1995, and I think probably one of the first of its kind in Australia. And so I came back to Sydney. We'd actually just moved to the new Children's Hospital. For the first time at this hospital, there was a research building, and there was 10 of us in that building when this hospital opened. And now I think there's probably about 600 researchers there today. But so I came back from this amazing place in Boston to an empty lab in a virtually empty research building in this hospital in Sydney. And I must say I was clinically depressed for about 9 months, because instead of having lots of people to engage with and to help me, I had to set it all up on my own. And the way I got through that was to think, I'll give this a good go to see if I can run a laboratory and to build it up. But I'll give myself 2 years. And if I can't make a go of it, then I'll go back to what I find easy, which was clinical work. And I'm really happy to say that at the end of about 18 months into this, a neurology job came up at the Children's Hospital. And a lot of the clinicians said to me, "Oh, Kathy, your job's come up," because you had to wait for someone to die or retire to get a job in those days. And I didn't even apply for it. I thought, no, I found what I want to do. And I kept on, and I was doing a mixture. I still did clinical work, but this position, which I had for 3 years, allowed me to set up my research and to set up my laboratory, and I'm forever grateful for that. Meanwhile, Robert Ouvrier had charmed a donor, and he was set up an institute for neuroscience and muscle research. And at that stage, he was the director and I was the deputy director, but he basically set it up for me, I think, so that I could expand the research we were doing across not just the laboratory research, but how we take it into the clinic. So I set up a neurogenetics clinic at the hospital, and really the big focuses were, not surprisingly, my two areas of research interest, which were neurofibromatosis, which is really quite a common neurogenetic disorder, and we followed a couple of thousand families with that disorder. But in Boston, I'd worked very much on the causes of muscle disease and muscular dystrophies. So it was sort of a hybrid neuromuscular and NF clinic, and it still exists to this day. And since I've moved to Melbourne, I've set up an NF clinic down here as well. So that gave me the sort of opportunity to start to build. And this was my lab starting off this was by, the time we'd been going for a couple of years in 2000. And a really good mixture. Nigel was doing a PhD with me. He was a geneticist, and all the rest were scientists. We've got a genetic counsellor there, sort of a hybrid of people working together. And then by the time, 2010 had grown immensely. By the time I left in 2012, we had about 130 people associated with this research group. And again, as I look at this, is Leanne, who's actually up the back of the room. She's worked with me forever as my chief of staff and moved with me to Melbourne. There are a number of doctors, Russell Dale, who's a neuroimmunologist, and Christy Jones over here, who was my first PhD student. She now runs the neurogenetics service in Sydney. There are a whole heap of scientists, Monkel Leck and Angela Chen, and they also work with Daniel MacArthur, who was a PhD of mine. Daniel was in Boston at the Broad Institute for many years. Angela and Monkel are now at Yale University. Mickey did a PhD and she's now working in Germany. Pete still works with me at the Murdoch. I mean, it's a whole range. And then I had this great troupe of allied health professional researchers who did their PhDs, because working with clinical trials in neuromuscular disorders, you need that multidisciplinary team. So here we've got Josh Burns, who's now the Dean of the Faculty of Health Sciences up at the University of Sydney. Christy Rose was a physio. Paula was an OT. Dave up the back was a speech therapist. So we sort of had this amazing environment of bringing together all of these academics across all of these disciplines to really focus in terms of what we could do, particularly for kids with neuromuscular disorders and kids with learning disabilities associated with NF. So let me just give you a flavour of what it was like doing research and why I've become really motivated to look at how we can bring genomics much more readily into clinical practice. All of these kids have neuromuscular disease. They all present with weakness or difficulty walking or difficulty breathing. But all of these kids have a different genetic basis for their muscle weakness, but it presents in similar ways. Now, this is the olde diagnostic flowchart of what we did back in the old days up to 2012. And very much after we'd done the clinical assessment and the very, very important family history, our first line of investigation was muscle pathology and muscle biopsy to look at the patterns within the muscle, which muscle is a beautiful thing. It's absolutely my favourite tissue. We'd do a whole series of protein studies and look for protein abnormalities. And then we'd be looking to pick the gene and we'd sequence them one by one by one. To study families, we then do linkage studies and we try and narrow down a region and narrow down a region. This stuff took years, as my good friends Chris and Ingrid will tell you, to actually find a gene and then find candidate genes that you'd sequence. And it was very long and painstaking work. When I started my lab in 1996 and looking at all the various inherited myopathies, these were the genes that we knew about. But by using that ye olde process, by 2012, we'd really discovered and started to fill out this table. So we actually did quite well. But it was really very limited to where we could study large families or luck out and find really good candidate genes. So this Nemeline myopathy has been a disorder I've done a lot of work on, and the congenital myopathies are defined by what you see in the muscle biopsy. And this is a muscle biopsy with a Gomori trichrome stain. These are all these insertions are actually collection of the Z-line material. this sort of defines what nemaline myopathy is. Nema is Greek for worm or thread, and so you can see these little thread-like rods. Nemaline myopathy is a disorder, particularly of the actin thin filament and all the things that go to make the actin thin filament and that beautiful regular muscle structure. And you can see the rods actually arise from the Z line and consist mainly of a protein called alpha-actinin, which I'm not going to go into today, but is one of my favourite proteins. So this is Eliza. I looked after Eliza as a neurologist. It's her in the intensive care. She was unable to breathe off a ventilator until she was about 9 months of age, had generalised weakness. You can see the marked facial weakness that she had, and her diagnosis was nemaline myopathy. And you saw we had lots of genes for nemaline myopathy. So our workup was over a 5-year period. We did extensive protein studies. We sequenced all the known genes, and as we were discovering them, the nebulin gene was a really big gene. And to get that sequence, we had to send DNA over to America at a cost of $8,000 American to get one gene sequenced. But we were able to exclude it by doing much cheaper linkage analysis to show that that wasn't the case. And this was the pathology. It's a beautiful thing. I'm very sad we don't do many muscle biopsies anymore, where you're seeing this is beautiful normal muscle, and then what we're seeing in Eliza's muscle is these accumulations. This is stained with alpha-actinin to demonstrate the rod formation. And what was really interesting to me, probably as the only person in the room, was these rods. They were like little caterpillars. They had little hairs of thin filaments coming out of them. And that ended up being very important in terms of the pathology. So we had no genetic diagnosis after 5 years of investigation, but the parents decided to go ahead and try for another child. And it doesn't take much to tell that Sarah, the little Eliza's sister, had exactly the same thing as Eliza. She had nemaline myopathy. And so for this family, having these two kids that are severely disabled, I mean, it ended up there was domestic violence. The mum had to give up a job. The family split up. It was the stress is something that you often see of the impact of having, you know, a couple of severely disabled kids who are in and out of hospital all the time. So it can be quite devastating for families. But, you know, these were also bright little kids. Their brains weren't affected. Even though she had to hold a jaw up to be able to speak properly, Eliza decided to study Japanese and French. But then there came whole exome sequencing, where the technology had progressed so that we were able to sequence all of the genes at once. And in collaboration with Daniel and Monkel, who were my ex-PhD students who moved and were working in Harvard, we got early access to be able to do this type of genetic sequencing. And the first family we concentrated on was Eliza and Sarah's family. And I fell in love with genomics when the first thing came back, and it was really, we hit the gene really, really on. It doesn't happen that often, but it was just, we found a compound heterozygous variance in a gene called LMOD3, which hadn't been associated with disease before. And we confirmed that the parents were carriers and that the kids carried 2 copies of that abnormality. It was biologically plausible that this was a disease gene because this LMOD, there's only been studies on this other related protein, LMOD2. But what it did, it was really quite a fundamental protein that nucleated actin filaments so that they could grow and extend to form the thin filaments within muscle. Also think about back to that caterpillar rod. What you were seeing there is accumulation of Z-line tissue with little truncated bits of thin filaments coming out of them that couldn't grow properly because of the lack of lyomodin. You need to find additional families then, though, to be able to prove, you know, we couldn't just say this is the disease gene. And so at that stage, we did have email by that stage. And so in we didn't have databases to trawl through, so we emailed our mates, and we had great international collaborations around the world. And in doing that, we did genetic screening over a 6 to 9 month period of over 540 additional cases of Nemaline myopathy, and identified an additional 17 patients. And you can see from this table down on the left, it was from a range of countries in Finland, Italy, France, Japan, from around the world. And then the clinical features, in actual fact, our 2 patients were the only 2 to have survived beyond the first few months of life. And we actually demonstrated that that was because they still had residual protein that could partly do the job. But most of these kids, where it was lethal, they actually died early on in the newborn period, or were stillborn. And then the final thing we did was functional genomics, where we made a model to demonstrate that the mutation in this gene actually led to disease. And this is a zebrafish model, and you can see there's differences between the 2 at a whole body level. The control is on your left. But what we've importantly saw that was in the muscle there, that there was accumulation of these rods. This is staining with alpha-actin, and so we could reproduce this disease in an animal model. So that was great. And over the next couple of years, that was our first case, but over the next couple of years, we're actually up to solving 95% of Nemaline myopathy cases now through the advent of genomics. And previously, our success rate doing this would be less than 10%. So it's just a phenomenal step in a 3-year period to be able to give families an answer. And it also means now that we don't need to do muscle biopsies as a first-line investigation with these kids. And meanwhile, the cost of doing sequencing has come down dramatically, and the technology of the bioinformatics and the computer grunt to deal with the 3 billion base pairs as part of analysis has also amplified. So I loved my job in Sydney. I had a wonderful group of people. It was all humming along greatly. I had tenure as a professor, but I still thought, is there something more that I want to do? And at this stage, I was approached about the job that was coming up as director for the Murdoch Children's Research Institute, and I think you all know the end to that story. So in 2013, I headed down to this amazing campus where there is an integration. What really attracted me was the integration of the hospital, of the Medical Research Institute, where we have 1,300 researchers. We have 40% of our research groups are led by clinician-scientists. So it's a real hotbed for clinician science. And we're also with the University of Melbourne Department of Paediatrics. So it means that it's a perfect environment for you to see a patient in the clinic and then take the questions that arise in clinic into the lab to answer them and then back out again, which I think is incredibly important from a translational perspective. The other thing that attracted me is that the Victorian Clinical Genetics Service, by quirk of history, is a wholly owned subsidiary of the Murdoch Children's Research Institute. So there we had this environment where if we could take our genomic discoveries directly into the diagnostic laboratory. And so it was at this stage, I sort of changed direction a bit. I mean, I still run a lab and still quite personally active in research, but I thought it's great if you happen to stumble on the clinics where it's associated with great clinician researchers who will really go all out to find diagnoses and to present you with the latest treatments. But how do we take that to scale, and how do we provide equity of access around the country? Genomics, I think, I don't need to tell all of you guys, from a diagnostic perspective, is amazing. And so what we did from a genomics perspective is we set up a partnership and network around the country that Chris mentioned called Australian Genomics, which now is continuing to this day. It's been going for 6 years now, with 100 partner institutions around the country, as well as major international linkages, and over 450 investigators around the country. And the goal of this is to demonstrate the value of taking genomics into clinical care and the practical strategies to do it, because we actually have a disruptive technology that we need to move into the health system, and we need to do that in an effective way and create genomics literate clinicians. And I would say to all of you, wherever you are in your career, you're going to need to have a good working knowledge of genomics. My dream also is that from a research basis, that Australia isn't fragmented and siloed, that we function internationally as a single cohort of 25 million people. And we're achieving that from the research perspective in terms of creating national networks, and it gives us a much greater standing internationally. Chris also mentioned my association with the Global Alliance of Genomics and Health, and this was actually pushed for by Francis Collins, who led the Human Genome Project, with the goal that, okay, you've got the human genome, what are you going to do with it? We need to be able to make this accessible in clinical care so that you can pick up your phone and your computer and you can find the evidence for a specific genetic variant really easily so that you can apply it for your patient or to your research. So the Global Alliance is now involved over 90 countries, and it's driven by real-world programs of work, of which Australian Genomics is one, and Genomics England. And we really interface with the expert geeks around the world to solve the problems in terms of how we capture and interpret and share genomic information. So it truly is a global organisation, and really what we're aiming to do, and where you see the little Global Alliance logo, is where we've created the standards or the tools for every stage along this pathway, from patient to research into healthcare. So what have we done in Australia? What have we achieved? Because it's, none of this has got any point unless you're actually changing clinical care. But we now have, we've now found that across a whole range of rare disorders, by applying genomics early on in that diagnostic odyssey, not having to do muscle biopsies, for example, that we can provide 5 times the diagnostic rate at a quarter of the cost of the typical diagnostic workup. We've been able to start working in intensive cares with critically ill kids, and we started this as a pilot program in Melbourne where we could get the time to diagnosis from 6 months down to 4 months, down to 1 month, down to 2 weeks. And now we've got a national program where we can do it in 3 days, so that if there's a critically ill kid who's thought likely to have a genetic disorder, we can do that rapid turnaround. And that's meant that our diagnostic laboratories, they're no longer Monday to Friday, 9 to 5. They're now 7 days a week. The important thing there is we're hitting about a 50% diagnostic rate for these children that usually wouldn't get diagnosed during their time in ICU, and it results in a change in management in 75% of cases. And then there's Mackenzie's mission, which has been funded by the federal government. And this is really looking at how we can expand reproductive carrier screening. You can currently screen for about 3 disorders. We've actually taken this to initially a whole exome. We'll move it to a whole genome approach, where we can screen couples and look at their risks of over 1,400 genes associated with chronic and debilitating disorders, so that they have the choice, if they wish to take it, of preventing having a child with a severe disability. So it's everything going from, early intervention and prevention with reproductive carrier screening, but then moving into therapeutic approaches as well. So just as an example, this little boy was rushed to the ICU at the kids' hospital for really major hypertension and heart failure. But within 66 hours, this is one of our really good ones, so obviously we feature that, we delivered a genetic diagnosis of a very rare genetic disorder that changed his therapy and was able to control his cardiac failure and his blood pressure so that he develops now normally. And the other thing that we took all of this evidence and went to government and went through a very, very long process, but we've now got Medicare items for genetic testing for kids with both genetic syndromes and intellectual disability. And we've now got a pipeline going through to make sure that this testing is paid for as part of clinical care. So I'm going to jump through this quickly so that I can get to some really good advice to the end. But the other thing around the Murdoch is it's not just about genomics. There's a whole range of different areas that I've now dived deeply into and I'm quite passionate about, and particularly getting all of our different groups to crosstalk. We have great well in stem cell medicine, and we're using this to be able to model the rare genetic disorders in terms of providing models, human models of disease from pluripotent stem cells, but also being able to screen potential drugs. We do this across a whole different range of disorders, and Melissa Little, one of our superstars, is generating little mini kidney organoids and then developing kidney patches that can actually be transplanted into damaged kidneys that actually start to make urine. It's amazing. This is actually on the left here showing heart cells that have been grown from human stem cells, just from blood or from skin biopsies, differentiated into heart tissue, and then we can make three-dimensional structures that beat as they do, but can use this to then test drugs that will improve contractility, for example, for patients with cardiomyopathy. And we're now doing the same, I'm happy to say, with skeletal muscle tissue. And just last week, we created the first three-dimensional model from stem cells of Nemaline myopathy. And we can correct the gene defects, and so we can compare control and disease, and you can actually see the phenotype in the stem cells. So it's really very exciting. We also then think about population health because, you know, I've had a focus in my life on rare disease, but we then look at the modern epidemics, apart from COVID, that are affecting our kids. And so this is also a huge focus of the Institute, how we are reaching out and changing practice within the community across many, many common disorders. And we do this by, we've run and continue to run a number of large prospective birth cohort studies, studying everything from allergy to mental health, many of these now cross-generational. And they provide a wealth of information in terms of identifying the risk factors and what we should be doing in terms of early intervention. This is a study that's around Australia where we were actually able to identify early predictors of overweight and obesity, be able to predict kids from 6 to 7. If you didn't have those risk factors, your risk of obesity, was 13%. But if you had 3 risk factors, 71% of those kids were obese by the time they hit adolescence. So it's really picking the populations of kids where we need to intervene. Gen V, we're just rolling out across the state at the moment, is how we take that to scale. We're enrolling every baby born and their parents born in Victoria over the next couple of years. So it'll be around a cohort of around 150,000 babies and their parents, and collecting the longitudinal data that is collected but tends to sit in silos, the newborn screening cards, which we do at MCRI, and all the information that comes from the maternal child health nurses as to develop the state as a platform for going forward. I'm going to skip through, I hate to skip through immunity and infection immunity and global health, but we have reach and outposts in over 40 countries around the world. We've developed vaccines. This is the first baby in Indonesia receiving a rotavirus vaccine that can be given orally, can be kept at room temperature, which is so important in providing effective vaccines in third world countries. And then there was COVID. And what I was so proud of during COVID is that every aspect of the Institute turned their attention to what can we do. Our genomicists, we grew stem cell models of all the human tissues that were affected by COVID, and in partnership with the Doherty, we infected these and we've worked out looking at disease mechanism and again how to treat specific complications of COVID. We have an international trial that's looking at BCG vaccine, which is an immune booster in terms of protecting frontline health workers, particularly in third world countries. And this wonderful family is part of a large study that we've been doing to see why kids have been relatively protected so far in terms of COVID infection, and have demonstrated a different type of immune protection in those kids. So, what have I learned about being a clinician scientist? Let me leave you with a few pithy reflections. Obviously, I love research. It is been a part of my clinical practice, and I think the very best doctors are also researchers because we are thinking constantly of what is important and what is going to change practice for our patients, and we're doing evidence-based practice. There's an excitement about it. You get to do what you love to do. You've got to pay attention to the unexpected, and I see there's a talk later on the value of serendipity, and I can tell you there's certainly been bits of my career that were quite unexpected. There's a breadth of career choices across all of the different subspecialties, from doing clinical work, clinical trials, clinical research, to basic laboratory research. And it really increases, I think, the breadth of career choices that you have when you can, as well as your COVID vaccine passport. Medical research is the passport to travel because we become part of these amazing national and international communities that I really miss because I haven't seen them now for 18 months, like the mates that I emailed around helping me with anemoline myopathy in Eugene. And Australia is a great place to work. It used to be when I was young that you needed to do your BOSCB, which is you've been overseas, then you came back. But really now there's a very rich research environment in Australia, and we are really, we're out there and we're leading in so many areas internationally. It's an exciting time to be a researcher as well. There are all the new technologies, you know, the rapidity with which we can get, you know, genomics results, and we're starting to look at multi-omic approaches, and this is now becoming possible. And, you know, dibs to Francis Collins and the Human Genome Project, and I just love this quote, and this really signifies what the Human Genome Project has done. But now what we're doing with the Global Alliance is making sure that we're capturing and harnessing all of the genomes that are generated in both clinical practice and in research so that we can share that knowledge, because the more you've got, the more accurate you can be for the individual. So the positives first. Research is addictive. I mean, I just could not imagine not doing it. You'll never be bored. I hope I've conveyed to you that and you can change direction. You can pick different areas. Half of my institute never heard of COVID 2 years ago, and now we've yielded so much great research because people from all different areas pulled together, and it was exciting. There was a weird excitement about that experience, I must say, because we have a team of 150 researchers from population health, from global health and infectious disease, immunity, from stem cells, from genomics, all getting together and working together. It's really fantastic. You get to interest other people in research, and it's also, I think, very important to think that I think of research as fun. And when my people get agitated, particularly during the last year, I go, calm down, guys. It's only research. It's not life or death, but it is something that excites you and it drives you. And you've also got autonomy. You've got control over what you're doing. You can follow where you're interested. Having autonomy, I think, is incredibly important, and you've got to earn it. But I think it is really something that I value and cherish as part of my career. But research is hard. Your ego is going to constantly be challenged. You'll have papers rejected. You won't get grants. But I would say to you, it is such a rewarding career that it's not the time. There have been different times where I just thought I could go down this path and it'd be a lot easier. But I actually find that I always think about that path of most resistance rather than taking the path of least resistance, because it's more likely to be rewarding for you. Finally, pursue your passions. You've just heard in the videos before about so many of us, and I can speak for, with knowing Ingrid and Chris well, of people that have very single-mindedly pursued their passions. Have the courage to make those difficult choices that might be the easy way. And I think most importantly, life is too short to work with difficult people. You will find them along the way. There are a lot of people that are possessive and don't share. Don't work with them. It's not worth it. It's no fun. And I think that's probably, I wish I'd learnt that earlier on in my career, rather than trying to persist because a certain collaboration seemed like it was going to be really, really useful. So with that, I will come to the very final point, which is the best part of being a researcher is being able to interest other people in research and to help guide their careers. And I love to do that with people at all different levels within my institute. It gives me the greatest of pleasures. Also, you are young people and you are looking for mentors, but there are a lot of younger students coming behind you, and for you to take other people under your wing and to guide them along the way from your experience is also very important. I talk to my young postdocs and say, be really nice to the students that are coming through doing placements, because the people I remember are the people that were really, really nice to me when I was a student, and you want to be that person. And also those people get older, so if you weren't nice to them, they'll probably be employing you one day. So it's always a very good advice. And I'll leave it there.